Hello, my name is Patrik Brundin. I am at the Andel Institute in Grand Rapids, Michigan, and I'm going to ask a question here that I hope to answer during this talk. Can infections be a trigger of MSA? First, just very briefly, I have some disclosures to make, company affiliations or commercial support or ownership interests, and here they are. My presentation today is divided into two parts. I'll start talking about synucleinopathies. What are their similarities and differences? I'll explain what that long word means in a second. And then in my second part, the main part of my talk, I'll discuss infections as a potential MSA trigger. Now, when it comes to synucleinopathies, those are diseases where the protein alpha-synuclein adopts an abnormal shape. And there are different conformational states that alpha-synuclein can adopt. And you see this picture, it has three parts, the blue part, the yellow part, and the red part that are biochemically distinct. One likes to bind to lipids. The yellow part in the middle, on the other hand, likes to aggregate. And the red part to the right on your picture has an ability to bind metals and other proteins. Normally, alpha-synuclein doesn't have a very clear structure. It's what we call natively unstructured when it's in solution, except when the lipid binding part sticks into a fatty lipid membrane. But those natively unstructured synuclein molecules can associate with each other, so they come close to each other and form what we call oligomers, or partially folded nucleating structures first and then oligomers. And oligomers are several synuclein molecules that come close to each other and interact. And they can do this in a variety of ways, some which we call off pathways, and we don't really know what they end up doing. And then there are those that we call on pathway oligomers. And they tend to associate, so there will be many, many synuclein molecules together, so many that they form what we call protofibrils. These are difficult to dissolve for the cell, and eventually the protofibrils in turn will assemble into even larger structures that we call amyloid fibrils. There are different types of structures of amyloid fibrils, different types of long chains of multiple synuclein molecules, and we call them strains. Strain A here, strain B, strain C. And we have learned over the past 10 years or so that they will associate and be present with in different brain diseases. And therefore, if you look at this slide, we have the rare disease MSA and the two relatively common diseases, dementia with Lewy bodies and Parkinson's disease, that share a certain feature, which is, they're all synucleinopathies. The feature that they share is that synuclein forms aggregates in this disease. As you can see, the brown coffee bean shaped cartoon. Now, if we look at this more carefully, we will see that the coffee bean shaped aggregate ends up in different cells. In MSA, it's in the red cell to the left, and in Parkinson's disease and dementia with Lewy bodies, it's in that star-shaped nerve cell to the right. I'm going to explain what that is a little bit more in a second. But first, I want to focus on Parkinson's just for a few minutes, which is the first synucleinopathy that we realized existed in terms of synuclein playing a role. And the history goes back to 1912 when Louis, the guy circled in the photograph there, discovered that there were strange clumps inside nerve cells in Parkinson's disease. He actually worked with Alois Alzheimer, who of course gave the name to Alzheimer's disease in Germany. Now let's fast forward from 1912 to 1997, when Michel Goerdert, working with his wife Maria Grazia Spillentini and the research scientist Ross Jakes together found that alpha-synuclein is the main constituent, the main protein constituent of Lewy bodies. This was a huge discovery because suddenly we knew what was inside those protein clumps that had been known for a long time. And if you look to the right, these Lewy bodies could look different. sometimes in the outer layer of the brain, in the cortex, they'd have a certain shape. In the basal parts of the brain, they'd have a different shape. Sometimes they would be inside the cell body, and other times in the processes of the nerve cells. So what happens early in Parkinson's disease in terms of symptoms and Lewy bodies, or Lewy pathology, which is a name that summarizes the aggregates in cell bodies and those in the processes? Well, when it comes to symptoms of Parkinson's, you all know that there are motor symptoms, problems moving, but interestingly, long before they occur, almost all patients report one or more non-motor symptoms. And circled in red, you see the phase here of non-motor symptoms before the diagnosis. And they include things such as depression, anxiety, importantly, constipation, loss of sense of smell, and sleep disturbances. And the Lewy body that you see in this picture, that type of pathology isn't just found in parts of the brain that govern motor symptoms. And in this rather complicated diagram that I'm showing at the bottom here, a scientist called Heiko Braak and his collaborators divided different stages that Lewy bodies appear in, different stages of the disease where they are, into 6 different levels or set stages. And in the very first stage of Parkinson's, the Lewy pathology is only in a couple of brain regions, and one is related to the sense of smell. It's the olfactory bulb. The other one, which is here labeled dorsal motor nucleus, or the vagal nerve, it has the Roman numerals down there, that is a part of the nervous system that governs movement of the bowel. Hence, it could be involved in constipation. It's inside the brainstem. It has a very long nerve fiber that innervates the gut. As the disease progresses, other parts will be engaged, like the locus coeruleus you see here, which could potentially be involved in sleep, wakefulness, and sleep disorders in Parkinson's. And we have now pathology in several brain regions that are not involved primarily in motor function. It's only when you reach stage 3 or 4 that the part of the brain that everybody associates with Parkinson's disease, the substantia nigra, gets Lewy bodies. And then later in the disease, you'll see on that brain to the right, it's red all over. Those Lewy pathologies are all over the brain. What does this mean? Well, Heiko Braak suggested that the Lewy bodies start in 2 parts of our body, the nose or the gut. And at the time, he didn't know what the Lewy bodies, how they moved around, or if they could move around. He thought maybe it was a virus that was traveling along nerve cells, along the pathways, and then infected the next cell. Today, we think, we don't know for sure, that the infecting agent might be actually the Lewy bodies, or fragments of Lewy bodies that move from one cell to another. And then you get this progressive involvement of several parts of the brain. This section here in this next slide shows the type of alpha-synuclein pathology that one primarily see in MSA. And here it's different because it's not primarily in the nerve cells. It's mostly actually in supporting cells called oligodendrocytes. So in this cartoon, you can see on the left that MSA primarily has the red cell involved, the oligodendrocyte, which is a supporting cell that forms the insulating myelin sheath that surrounds the long axonal process that nerve cells can have. And that's where the pathology seems to begin in MSA. On the right-hand side, you see that the Lewy body is actually inside. That coffee bean shaped is inside the nerve cell and somehow probably is involved in killing the nerve cell. Why am I telling you all of this? Well, if we think that the pathology starts in the early stages of Parkinson's disease in possibly the gut and the nose, where would it then start possibly in MSA? Well, let's look at the symptoms of MSA in this slide and how they evolve over time. There is a phase that we can call pre-motor MSA, when there are no problems moving, and they involve a variety of non-motor symptoms. Some are similar to Parkinson's, There can be REM sleep behavior disorder, for example. Actually, sometimes loss of sense of smell, but there are some things that are a little bit different to Parkinson's disease, and let me emphasize that. And let me go to the next slide and describe to you an idea that maybe pathology spreads from one brain region or one body region to the next also in MSA. And you know, there are two major types of MSA, the one called MSA-P for Parkinsonism and MSA-C for cerebellar symptoms. Two different patterns of pathology with different types of motor disturbances. Now, if we come back to the picture showing the timeline, you see that in the middle of this picture is when the Parkinsonism or cerebellar features appear. But before that, in the red box, some of the earliest signs and symptoms of MSA, are sexual dysfunction and urinary dysfunction, such as incontinence. And quite frequently, there are urinary tract infections. So in the next slide, I summarize the different typical features in MSA-P and MSA-C, and how they then potentially, if you look at the lower part of the slide, how they might relate to what parts of the brain that are involved. And there is a correlation. So the patients with MSA-C, they have obviously the cerebellum involved, and the patients with MSA-P have the bits that are also involved in Parkinson's disease are affected. But as you see in the next slide, what might be most interesting when trying to understand the origins of MSA is to look at the peripheral nervous system. And we look at the peripheral autonomic nervous system, and we look at also at the central nervous system, the brain and the brainstem, and how they might interact. Now, all the symptoms that can occur in the periphery are very interesting in MSA. They can affect the heart, the lung, the nose, the gut, but in particular, as you see in the bottom part of the left part of this diagram, we look at the urology, not the neurology with an N, but the urology with a U, which involves sexual dysfunction and urinary tract dysfunction. These are very prominent features, and they typically precede the classic features of movement disturbances. And the fascinating thing is, a Chinese study about a year ago demonstrated that MSA, but not Parkinson patients, have alpha-synuclein pathology in the urinary system. So they took biopsies in the urinary bladder or in the sphincter, the muscle that stops the urine from coming out until you want it to come out. They took these biopsies and they stained it for alpha-synuclein aggregates. And those brown and blue pictures to the left actually show what you see in the microscope. And to the right, you see where the biopsies were taken. And in the middle, you see bar diagrams showing how many patients actually had pathology in these muscle areas. And MSA-P and MSA-C patients both had pathology in this area, but Parkinson's disease and a disease called progressive supranuclear palsy, abbreviated PSP, and normal controls, none of them had pathology in these areas. So it seems that we've identified a unique anatomical pathology feature of MSA in the periphery. And that's why I'd like to go into the second part of my talk, which is, could infections actually be a trigger, an initial trigger of MSA? And this is super interesting because it's not something we traditionally would imagine. So here we come back to the two circles, looking at the trigger and asking, how does it get to the brain? Well, it has to go via the periphery. If it's an infection, we don't think the infection affects the brain primarily, but something in the periphery. So about 3-4 years ago, we started exploring if UTIs, which stands for, of course, urinary tract infections, could trigger alpha-synuclein accumulation. And we've done a lot of experiments. This is work done by a very talented postdoc, Wouter Perlitz, who's also in this conference. And it says this work has been submitted for publication. It's currently being reviewed by a scientific journal. They've given us some feedback, and we're working on their feedback, hoping that we can publish this in a few months from now. What we did is we generated urinary tract infections. We had to get some new collaborators. We are neuroscientists. We work at the other end of the mouse, in the head. We're not used to the urinary organs, but we got our collaborators and we worked together on this. And we showed that when we infect the animals with what's called UPEC, uropathogenic E. coli, a bacteria that likes to attack the urinary bladder, then, as you can see, we get total alpha-synuclein, T-acin, increases dramatically, and I-alpha-synuclein is insoluble alpha-synuclein, that's aggregates. That goes from almost 0 to very high levels. And when we look in a microscope, we can see in the picture, which is labeled ACE in red at the bottom there, that we see alpha-synuclein, and it's co-localized with a protein called MPO. I'm going to introduce that in a second, why that's important. But at the bottom right, we can see that MPO goes up at the same time as alpha-synuclein goes up. The more colony-forming units of bacteria we put in the bladder, the more it goes up. So it looks like MPO and alpha-synuclein go up together. But why we're interested in MPO? Well, myeloperoxidase, which is what MPO stands for, is a protein found in white blood cells. It's a protein found in particular in neutrophil leukocytes, and we therefore became very interested in these white blood cells. It seemed like alpha-synuclein went up at the same time as white blood cells invaded the urinary bladder that was infected. And in this next slide, you can see that neutrophils can do or form something that's called neutrophil extracellular traps. It's abbreviated NETs. And we became very interested in these because we got a hint that maybe the NETs that they form, which are these masses of molecules that can trap bacteria, may actually also contain alpha-synuclein. In the next slide, you see some pictures from the microscope where we looked at human neutrophil cells, so white blood cells from humans, and we could see alpha-synuclein outside cells that were outside these neutrophils when we challenged them with UPEC, with a bacteria. And in the other one on the right, you can actually see tissue from human urinary bladders, where we also see alpha-synuclein outside white blood cells. We label the white blood cells with the MPO marker. So human white blood cells form these nets that contain alpha-synuclein. It surprised us. We weren't really aware of this, and not many scientists had pursued this. We went on to look more carefully in the next slide at these particular nets, what they looked like, and we labeled them with something called H2B, which made us certain that these were nets. This is a histone which is present on the DNA of cells, and it was just a way for us to confirm that these were really those nets that the cell was casting outside. So to study this even more, in the next slide, we started to genetically engineer cells. And we took a cell line that's called HL60, which is like a neutrophil. And we put inside this, with genetic technology, alpha-synuclein that had been merged to a yellow shiny protein. YFP stands for yellow fluorescent protein. And we made lots of different cells of these types. And we had those that had lots of alpha-synuclein with the yellow label, and those that had a little alpha-synuclein. And then we put them in a machine, and we took pictures of these cells overnight for 24 hours, every 15 minutes, as they were growing in a cell culture dish in the machine. And we added bacteria. And this movie on the next slide shows what we saw. The yellow fluorescent protein, I think you can see. The blue just as a way for us to label cells. And imagine that this is actually filmed overnight. So it's pictures taken every 15 minutes as these yellow cells are moving around and navigating amongst bacteria. Occasionally you see clouds form. As the movie's running, there are yellow bursts coming out. Those are nets being cast, trying to kill bacteria, and they include alpha-synuclein. So in the next slide, I can summarize what we've learned so far. We've learned that bacteria can trigger alpha-synuclein expression, which means that alpha-synuclein goes up. We saw that in our first experiment. It's very low under basal conditions. It can cause aggregation, so it will form clumps that are insoluble, not too different to what we see in the brain in MSA. And those clumps can be released by neutrophils. So in the next part of our experiment, we wanted to ask, could those clumps move from the periphery to the brain? Could alpha-synuclein pathology end up in the central nervous system? Could it even replicate some MSA features? And I told you, as you see in this next slide, that alpha-synuclein is expressed in oligodendrocytes in MSA. And we now had to create an animal model where we had alpha-synuclein in oligodendrocytes, and where we had these types of fibrils that I introduced you to in the beginning that alpha-synuclein can form. So in the next slide, I'll show you that you can build fibrils in a test tube over time. You can start with individual units of alpha-synuclein, monomers. You can make them misfold in the test tube. You do this over hours or days, and then they'll elongate and form insoluble fibrils to the right in this diagram. So we set about doing this. And we had a collaborator, or we still do, Magdalena Ivanova at University of Michigan. She took tissues from brains of people who had passed away with MSA, extracted fibrils from these, and then ran them through some fancy test tubes, and was able to build more and more fibrils in test tubes. You can see the MSA patient, the red line here, and there were no fibrils generated when she took tissue from control people who didn't have MSA. So she could produce those gray fibrils at the bottom part of the slide here in a test tube. And they were very, very similar, if not identical, to those found in the brain. And we took them, as you see in the right part of the diagram, put them in a syringe and injected them into the urinary bladder of mice. But we didn't just take any mice. So in the next slide, you will see that we used a genetically modified transgenic HU stands for human mouse that had human alpha-synuclein. Because we were working with human MSA patients, we wanted a humanized mouse. Only this protein was human here. And Wouter will talk about this in this conference, or has already done so, that he's generated a specific type of humanized mouse where he uses viruses to express alpha-synuclein in oligodendrocytes, which is the cell type that's most affected in MSA. So here we had the perfect model system, and here we took our fibrils that Magdalena Ivanova had made in a test tube at the University of Michigan, injected them into the bladder of these mice. We followed them for 9 months after fibril injection. And at the end of that time, or during those 9 months actually, we looked at the animal's ability to walk on this red track that you see here, and we traced where they put their paws, how heavily they pressed onto the surface. Those are the green labels there. It's left foot, right foot, and it's hind limb and forelimb. So we were able, in a very fancy scientific fashion, to see if they had normal gait. And we actually saw some changes in their gait. So over time, after injecting fibrils into the urinary bladder, something went wrong with their ability to walk. And then at the end of the experiment, we took the animals and, as you see in this schematic, sectioned the spinal cord and the brain stem and looked for pathology. And here in the next slide is what we found. It's a complicated slide. To the left, you can see a brown staining. That is alpha-synuclein pathology. To the right, you can see bars showing where we found it, in which mice we found it. We had all sorts of controls here, but the bottom line is, in the cervical and the thoracic parts of the spinal cord of these animals, we found lots of pathology if we had injected fibrils in the urinary bladder. So this suggests that the pathology could propagate, could move from the nerve cells that innervate the bladder into the spinal cord, and then from one nerve cell at the bottom of the spinal cord to one much higher up. And this coincided, you see at the bottom part of this slide, with the development of inflammation. So EBA1, IBA1 is a marker of inflammatory cells. And only in animals where we had those fibrils injected in the urinary bladder did we see inflammation going on in the cervical and in the thoracic parts of the spinal cord. And we're continuing to analyze these animals, and we're seeing pathology in other parts of the brain. And we think this is a slowly progressing disease, not too different to MSA. So, do urinary tract infections trigger synucleinopathy, pathology with these synuclein aggregates? Via an innate immune response. That's a fancy word, but it's another way of saying via white blood cells in this case. Well, we know that the infections trigger alpha-synuclein upregulation and aggregation. That's the earlier experiments I showed you in the mice where we looked at the amount of synuclein we got and how it formed insoluble clumps. We know the second line here that the neutrophils, the white blood cells, can express and they can release alpha-synuclein in those nets. And I've shown you videos of that. We've traced those with the yellow marker. And we also know that when we inject MSA fibrils that were made from patients, the brains of patients with MSA, when we inject them into humanized mice, into their urinary bladders, we can get the pathology to move from the urinary bladder into the spinal cord, up the spinal cord towards the brain. This is actually quite similar to what one sees in MSA. And here in my final summary slide, the question is, what are the origins of MSA? Well, our hypothesis, it's not proven, but it's still an idea, and we're getting more and more support for this, is that urogenital organs are affected early. Well, that's, I think, a fact, but could be a starting point for MSA. We're not thinking it's in all cases. We think some of the differences that one sees in the clinical course of the disease and might be related to where the infection is, but we believe that in some patients, maybe this is a starting point. Urogenital dysfunction is very common as an early feature of MSA. And synuclein pathology occurs in the urinary tract, as shown by that Chinese group that I showed you slide from before. That doesn't happen in Parkinson's. We know that urinary tract infections are frequently associated with MSA. Could be a cause. It could also, of course, be a consequence of MSA, but it's an interesting thing. And we know that urinary tract infections are also associated with higher levels of synuclein levels in our mice, and that the white blood cells, the neutrophils, can release synuclein in these extracellular traps. And finally, that the aggregates can propagate, as I just showed you in the mice, from the urinary tract to the central nervous system and up the central nervous system. So with that, I'd like to thank all the people who've worked so hard on this project and still are working very hard on this, and the people who've helped fund this work. And you can see a little picture there of Wouter Pelitz, who has been the lead postdoctoral fellow in this project and has worked very energetically. And with that, I'd like to thank you all for listening. And I'll welcome questions when we have our Q&A sessions.